Thank you all for joining us. I'm Patty Stoll, Director of the Women's Brain Initiative, and we're delighted to have Dr. Andrea Gore here as our speaker for today's seminar series. Just a couple of housekeeping items per usual. We have 2 microphones that will be available for the Q&A, so we just ask that you ask the questions into the microphone so that we can video record it. And also, we did leave on your chair a piece of paper that tells you what's coming next. David Page will be here in June. And then we also have a date for the symposium coming up. So we hope that you will mark your calendars. And then with that, Dr. Ursula Kaiser is kindly hosting Andrea's visit today to the Brigham. And so she will do the introduction. Thank you.
Thank you all, and I'd also like to thank all of you for coming, and I'm just thrilled to have the opportunity to introduce Andrea for her talk and to have her here visiting. We just had a great day, and we really enjoyed having a little bit of time with Andrea today to discuss some of our work and get her input. So it's really fantastic to have you here, Andrea. And Andrea is a long-standing friend and colleague who I've known for many years, so it's very special to have her here. So Dr. Andrea Gore is professor and Vacek chair in pharmacology at the University of Texas at Austin. Her research team is currently investigating how environmental endocrine-disrupting chemicals perturb the developing brain, sex differences in EDC actions, and transgenerational epigenetic effects of EDCs. So this is a very topical area, so it'll be really great to get educated about this area. She's been continuously funded by the NIH and NSF since 1995. She's published 4 books and over 170 scientific papers. She's received many honors, including election as a Fellow of the American Association for the Advancement of Science in 2008, University of Texas's Edith Clarke Woman of Excellence Award in 2016, and an Endocrine Society Laureate Award also in 2016. She's also very active as a mentor and advocate for the education of trainees. And she's been very actively involved in the Endocrine Society leadership, a society through which I've had the pleasure to get to know Andrea very well. She recently completed a 5-year term as editor-in-chief of their flagship journal, Endocrinology, and I had the wonderful opportunity to serve as an associate editor with her. So I can vouch for the fact that Andrea is one of the most conscientious, organized, and detail-oriented people I know. And in addition to that, at the same time, she has an incredible knack for looking at the big picture, and she's highly innovative and creative, both as an editor and a researcher. So I'm very thrilled that she's visiting us today, and I could tell you a little bit more about her research, but I think I will let Andrea tell us about her research. So without further ado, I'll turn the floor over to Andrea. I'm looking very forward to your lecture, and thank you again for visiting us.
Okay, I think I need to switch this over. There you go.
Good, okay.
Okay. Thank you so much, Ursula, for a wonderful introduction. It's really been great to be here and to get to spend time with you, as also I have to emphasize having you as a great friend and colleague as well, and of course, meeting new friends and colleagues. So let me jump right into the science now. I have a lot to tell you. This is going to be a rather data-full seminar. So what I've tried to do is summarize some data that have been published or are a little bit older and go through those rather quickly so I can focus on newer data and also some unpublished data. So you can see my title. I'm going to be telling you today about endocrine-disrupting chemicals, and I'm going to be calling those EDCs, and effects when we're exposed, as well as on future generations. So let me first set the stage for why I think this is an important topic. We've contaminated our world, and we can't uncontaminate it now that we've done what we've done. And so I just have a few little pictures to show you that remind me to tell you that here on the right you can see an example of chemicals that are just being dumped in the environment, and I study a class of chemicals that is exactly how they got into the environment, over the course of decades, and although banned for many years, they're now still found in the environment and in animals and people. The upper left reminds me to tell you that even in parts of the world that don't have much or any industry and are considered to be very pristine, people and animals, other animals, are still exposed to these chemicals because they're absolutely ubiquitous. They get around the world in water, through the food chain, through the air, and by many other routes. And finally, let me give you an idea of the scope of the chemicals that I'm going to begin to talk about. So here's a graph that began in 1900, goes through 2000, showing you synthetic chemical production in billions of kilograms per year. And of course, this ended 20 years ago, but that trajectory is continuing to go up. We synthesize huge amounts of chemicals, and it really began in the 1940s. I don't know if you can, let's see, I think this is, I'm not sure where the pointer is, but really began during World War II. And that was the chemical revolution, and it has just magnified ever since. That's okay, I don't need a pointer. Oh, it's, okay, I clicked on the wrong one. Okay, so of the hundreds of thousands of chemicals that we've manufactured, to date, about 1,000 have been shown to have endocrine disrupting effects. And I should note that thousands or tens of thousands of chemicals have not been tested for endocrine disrupting effects. So keep in mind the numbers may be much larger. The definition of an EDC that I take from one of the Endocrine Society statements I had the honor of being able to work on is an exogenous chemical or a mixture of chemicals that can interfere with any action or any aspect of hormone action. So here's how we're exposed to EDCs. They come from industry. I study PCBs, those industrial chemicals that have been dumped. Pesticides and fungicides, many of those have been tested and found to have effects on the endocrine system. Plastics, such as bisphenol A in water bottles or BPA replacements that now are in what we think of as the BPA-free water bottles, those contain some other bisphenol. Plasticizers, such as phthalates, which are found in medical tubing and food can liners, and many other chemicals. And this is just a subset of the sources of the EDCs. Now, we know that EDCs are in our bodies. There have been a lot of epidemiology studies. There's been a lot of work done by the Centers for Disease Control that have taken various samples from people from urine or breast milk or amniotic fluid or umbilical cord blood or fat tissues or other tissues, and they've assayed for these chemicals, and they're found in all of the different chemicals that have been investigated so far. And these EDCs are also very relevant to wildlife. Many populations of birds and fish and other animals have been really threatened because of the presence of these chemicals, and in particular, the reproductive toxic effects of some of these chemicals that prevent animals from reproducing and end up wiping out populations. So with that, by way of very brief background about chemicals in the environment and EDCs, I'd like to transition very quickly to the work that we're doing in my lab, looking at the effects of the EDCs on the brain in exposed individuals and across generations. And so the work that we're doing really is rooted in the basic principles of neuroendocrinology. I'm a neuroendocrinologist, where many decades ago, really a century ago, it was recognized that there are periods of development when the brain is particularly vulnerable or susceptible, either it could be in a good way or in a bad way, to natural chemicals that are involved in organizing the nervous system. And so in neuroendocrinology, we refer to brain organization as the groundwork by which the structure of the nervous system, the formation of synapses, neuron birth, neuron death, or the organization of neural networks, much of that happens very early in life, in the fetus, beginning in the fetus, continues early into postnatal life, and then ends shortly postnatally in rats. The timing is obviously a little bit different in humans and other primates. And this organizational period is a period when male and female brains develop differently, and we refer to that as brain sexual differentiation due to sex differences in endogenous hormones. Then later on in life, during puberty, as hormone release begins to increase from the gonads, the circuits that were organized early in life by hormones become activated. And at that point, behaviors associated with reproduction or other sexually dimorphic behaviors begin to be manifested. And so this concept really underlies all of the work that we're doing with endocrine disrupting chemicals, because we're interested in how exposure to these chemicals during this organizational period may change this developmental trajectory and lead to differences in health or disease, depending upon what and when that exposure was. And so this really is part of the field of DOHAD, or the developmental origins of health and disease, whereby the seeds of disease may be planted very early in life, and only later in life, after some period of latency, are the consequences manifested. And so we've been asking the question of, how do EDC exposures during these early life critical periods change this developmental trajectory and lead to dysfunctions later in life? Because we study brain sexual differentiation, of course, we're also very interested in sex differences in these processes. And so throughout my talk, I'll be comparing and contrasting differences in males and females. Okay, so I think I've actually already made many of these points, but just very quickly. I think when people think about endocrine disrupting chemicals, they don't usually start by thinking about the brain. But the brain has among the highest concentrations of hormone receptors of any organ in the body. The brain can also synthesize hormones. It has the steroidogenic enzymes necessary to do that. The brain plays many functions that themselves are responsible for the regulation of other endocrine or reproductive processes. I study the hypothalamus, and so the endpoints I'm going to be talking about today primarily are social, sexual, and also anxiety-like behaviors. And again, there are sex differences in all of these properties of the nervous system. And so we've been asking the question of whether neuroendocrine systems may be vulnerable to EDCs. We use a rat model, and so we expose the animals to EDCs during pregnancy, look at the outcomes through life and through aging, and then to future generations as well. So just a little bit about the classes of EDCs that we study. And I'm going to introduce you to the first one here, and then I'll talk about the second one a little bit later. So when I began this work, I was studying polychlorinated biphenyls, or PCBs, because I grew up in New York State. I was doing work at Mount Sinai School of Medicine in northern Manhattan, and people in Manhattan were fishing in the rivers and eating the fish that was very highly contaminated with PCBs that had gotten into the Hudson River from industry that was upstate, in upstate New York. And so the population of New York City who ate fish out of the rivers really had high contamination, and the community medicine department was looking at these individuals to determine were there any adverse outcomes in pregnant women who were eating a lot of fish, and there were, by the way. So I got interested in the PCBs. We are still exposed to them today because they've gotten into the food chain. I'm going to talk about a mixture of PCBs, an industrial mix that was used for a long time called Arachlor 1221 or A1221. So on my figures, you'll see A1221 or PCB sometimes a bit interchangeably, but it's the same mixture of chemicals. The other thing I wanted to point out was the structure of PCBs. They are biphenyls. You can see there are 2 phenyl rings. They're chlorinated at some variety of positions on those biphenyl rings. And although they don't look a whole lot like estradiol, that phenol ring allows the PCBs to interact with and bind weakly to estrogen receptors. So that probably is the main mechanism of action for PCBs. And so a lot of my work has compared PCBs to estrogens, and there are some similarities, but there are also some differences because PCBs are not pure estrogens and have other mechanisms of action. So here's the timeline for our experiment. We start with pregnant rats. During gestation, on day 16 and 18 of pregnancy, which is kind of the beginning of the third trimester of rat pregnancy, we expose the mom to the PCBs or to the vehicles. We have a vehicle control that's DMSO in oil, or an estradiol control to account for potential estrogenic effects of the PCBs, 2 doses of the Aerochlor. We allow the animals to grow up, and then we do phenotyping in adulthood of behavior, changes to the nervous system, protein, gene expression. And I should emphasize, these are within the exposed animals. So the only exposure to these animals was the treatment on day 16 and 18 of pregnancy. After that, there's no more treatment to the animals, and then we're looking at these animals are our subjects to do the phenotyping later. So I'm going to summarize these data very quickly. I know I told you there are going to be a lot of data, and there's a lot of data just on this one slide. But the point that I want to make with this slide is that we've looked at an expression of a number of proteins through immunohistochemistry, including ER alpha, ER beta, kisspeptin, and GnRH in the hypothalamus, all important molecules for the control of reproduction and behavior. And we were able to show that there are sex differences in expression of these molecules that we looked at, and that when we expose the animals prenatally to that very short period of EDC exposure, it decreased ER alpha, it decreased ER beta, it decreased kisspeptin, and it decreased GnRH neurons that co-expressed FOS during the preovulatory LH surge when GnRH neurons are activated. So all of these effects were found in the female rats. We did not look at ER beta in the males. We also did not find very large effects of the EDCs on male protein expression, but I should note other studies have looked at male brains and female brains, and it's just, I believe the 4 molecules I chose happen to be selectively affected in the females. Now, not everything changes, so we also did a really detailed analysis of vasopressin and oxytocin in a couple of hypothalamic regions with quantitative stereology and counting from rostral to caudal, and it turns out there weren't any significant effects of the treatment. So I like to show a little negative data because otherwise it seems like everything is always affected, and whatever I look at, there's going to be a positive outcome, and in this case, this system was not vulnerable to the endocrine disruptors. We've done a tremendous amount of work on gene expression in the hypothalamus of the animals in adulthood, and this shows a subset of the data in adult female rats. In this case, it was in the medial preoptic area of rats, of females. Let me orient you, because I haven't shown you any bar graphs, really, or I haven't talked about any bar graphs yet, but they're all oriented the same way from left to right. The vehicle group on the left in white, the estradiol group in light gray, the lower dose, 0.5 mg per kg of Aerochlor in the darker gray, and the higher dose of Aerochlor 1221 in black. So it always goes from lowest to higher doses from left to right. As I said, this was just in the females. And if you look at the genes, what you can see is that there were many genes whose expression was affected by the Aerochlor. Interestingly, most of them were upregulated. So for example, ER alpha was upregulated in both of the Aerochlor groups, notably not in the estrogen group, but so that implies there probably was not an estrogenic mechanism. ER beta is upregulated in one dose group, but not the other. Kisspeptin, here's kisspeptin. This lower dose of Aerochlor actually had more effects than the higher dose, and this is pretty consistent with effects of hormones and endocrine disruptors which have non-monotonic dose-response curves. Sometimes lower doses have bigger effects than higher doses. If you're at the peak of an inverted U-shaped curve, you go to a higher dose and there's a lower response, and we see that all the time with this compound. So, and kisspeptin is upregulated about 8-fold here. We also, I didn't show you any of the data, but we also did this in males at the same time, and there were several genes that were also affected by the EDCs, but not as many as in the females. I'd also like to give you a little snapshot of some of the behavioral data that we've done, because what I've shown you is that certain proteins and certain genes in the hypothalamus are affected by prenatal exposure to the EDCs. But for me, the important question is there a phenotypic outcome on, let's say, reproductive physiology or reproductive behavior or other types of behaviors? So before we euthanized the animals, we also began running them through a series of behavioral tests of social and sociosexual behavior, vocalizations, which are the way that animals communicate or that rats communicate with each other, mating tests, and some tests of anxiety-like behavior, like the elevated plus maze or open field or light dark box. And again, I'm going to show you just a little snapshot of data. One of the tests where we saw significant effects of the prenatal EDCs consistently in both males and females was a test of sociosexual preference. So let me play this little video. And what's shown is a female rat in the middle running around. That's the animal that was prenatally treated with the EDCs. And in either corner is a male. She's never met either one of these males before. One of them has they're both castrated. One of them has testosterone replacement and the other doesn't. And so typically, animals will choose to spend time with the hormone-treated animals over the non-hormone-treated animals. And we can record a large number of the behaviors, the time they spend, where they spend it, how they interact, how they touch noses when they're sniffing across that little barrier. And at the end of the test, you can see we end up with this plot that shows us where the animal spent all its time, and you can see this rat spent a lot more time in proximity to this rat than it did to this rat. So I'm going to show you one behavior that was affected, in this case more so in males than in females. This is when the animals are touching noses, which we think is a very important way that animals assess the sort of relative value of another animal as a mate. And what normally happens, for example, in this vehicle male, is animals will spend more time near the hormone female than the no hormone female. So black is hormone and white is no hormone. And we see that same pattern here with estradiol and with the higher dose Arachlor. Actually, the pattern is completely gone in the same group where we see most of the effects in the females. And overall, nose touching is decreased in any of the treatment groups compared to the vehicle. The females just don't nose touch very much, so that's probably why we don't see any significant differences. Just not enough data points. I'd also like to show you one other behavior, because this is not looked at by too many people. And these are measures of ultrasonic vocalizations, which are the mechanism by which animals communicate with each other. And we used a test where we would take two animals. One would be, let's say, a female treated from the EDC treatment, and we would put her in a cage with a barricade, and on the other side of the barricade would be a untreated male, just a normal gonadally intact male. We would let them interact across the grid. You remove the male and you record the vocalizations, and the females call a lot when you remove the male, and that way we can record them when they're by themselves. If I recorded them when they were together, I wouldn't be able to distinguish the female from the male calls. And we can do the same experiment with the males, where we introduce them to a female, let them interact, remove the female, and measure his calling. And so what's shown here is the calling after the stimulus animal has been removed. with females on the left and males on the right, same treatment groups. And what you can see is that for there are different types of calls. Actually, I can play one of them for you. This is a flat call. Oops, that didn't work very well, but it's a very flat tone. And these calls are thought to be indicators of different social states, although we don't necessarily know the difference between all of them. But what you can see is that there was no effective treatment on any of the flat calls. There was an effective treatment on rise calls in males and on step calls in both males and females, although very different. In males, calls in the Erichlor groups are increased. In the females, calls in the estrogen group are decreased. So we have sex differences in the effects of these prenatal treatments. And then finally, we've also, just as an example of some anxiety-like behaviors, we ran our animals in a light-dark box. They prefer to spend their time generally in the dark, and so we can measure their anxiety state by looking at their crossing in between the light and the dark box and where they spend their time. And again, in this test, only males were affected. So the speed of the animals, the time they spent immobile, crossing lines, and the time in the light box were differentially affected by the treatments, but not in the females. So to summarize this section, which is the background of the effects of the prenatal EDCs on adult behavior in the exposed individuals, these data, which I went through rather quickly, suggests that there is a network of specific genes and proteins that are involved in the hypothalamic control of reproduction and neuroendocrine function that is reprogrammed by these prenatal EDCs. We see differences in neural circuits, we see changes in gene expression, and we believe that that is, when measured in adulthood, and we believe that indicates that some sort of programming happened earlier on that has now changed their behavioral outcomes. I pointed out once or twice that the effects of the PCBs differ very frequently from those of estrogen, again, underscoring the point that EDCs are not like drugs. You're not giving an animal an estrogen when you give it PCBs. You're giving it a chemical that can act on estrogen receptors, but can also act on other receptors and other systems altogether that may also be important for the behaviors that we look at. And so the behaviors that are involved in reproduction, social behavior, communication, and affective state are perturbed by the prenatal EDCs. So to conclude this section, I feel pretty confident in saying that prenatal EDCs have latent sexually dimorphic effects. I should also point out that most of the effects I've shown you are for behavior in the males and for gene and protein expression in the females. And I should note we do see some behavioral effects in the females and some gene and protein effects in the males, but that actually is the pattern that we tended to see more often than not. So clearly there are other genes and proteins involved in the behaviors that we would need to look at to try to get a relationship between what's going on with neural circuits and behavioral outcomes. Okay, so I wanted to spend the rest of my time talking about multi-generational EDC effects. So let me kind of take you through this diagram of a pregnant woman to make the point that if this woman is exposed to an EDC, she is exposed, her developing, I ignore the baby who's feeding out of the bottle, but her developing fetus, that child referred to here, F1, this is also an F1, but in utero F1 is also being exposed. So the animals we were looking at before are equivalent to this child being exposed. Presumably the EDCs can get into the brain, have actions there, and also get into the brain of the mother and the body of the mother and other parts of the fetus and have indirect effects on the developing brain of this individual. However, it's important to point out that this exposure also affects directly the grandchild, the F2 generation, because within the F1 fetus are the developing germ cells, which will become the sperm and the egg cells. And so these cells have actually come into some contact, maybe limited contact, but contact nonetheless, with the endocrine disruptors. And so there's been a little bit of research, and I'll show you some research on what is going on within the F2 generation. But I also need to point out that we have on this slide a great-grandchild as well. And so I'm going to talk, when I show you the data, you'll see there are effects on the F3 generation of endocrine disruptors. And what this implies is that information from these previous generations and presumably programmed into the germ cells of that will be the F2 generation can be translated to the F3 generation. And we believe that there are epigenetic modifications happening within those germ cells that bring information to the F3 generation to cause a phenotype. So I'll show you some data to support that. Let me first just introduce you to the second class of EDC, which is called vinclozolin. Vinclozolin is a fungicide. It's used primarily in the grape industry, in the wine industry, for treating fungus in grapes as they're growing. And they're also used in sod, often used for golf courses. So we are exposed when we go play a round of golf or eat grapes or drink the wine, perhaps, that has come into contact with this EDC. And you can see the structure of vinclozolin. It also has a ring structure, which is quite common for EDCs. It's like Arichlor 1221. It's also lipophilic chemical. Excuse me. And the mechanism of action, again, it has probably a number of mechanisms, but we know that it is an anti-androgenic EDC and can act through blocking androgen receptors. So this is the rat model of what I just showed you for the human, and I realize this looks complicated. This was the simplest way that I could come up with to try to demonstrate the point that I want to make, because this will set up the experiment, because this really is a setup for the experimental design that I'm going to be talking about for the next little while. So this is a reminder that when you expose, in this case, a rat who's pregnant to one of the EDCs, the vehicle, the PCBs, or vinclozolin, you're exposing her, you're exposing the F1 embryo, you're exposing the F2 germ cell. And so the F1 animals, which were the ones I've been talking about up to now, are exposed in utero. Their brains are directly exposed. Now, what we do is we need to breed further generations, and the way we do that is we create either a maternal lineage or the paternal lineage. We do that by taking the daughters, the F1 daughters, breeding them with untreated males who never were exposed to anything. That leads to the F2 maternal lineage. And if we want to continue to the F3 generation, we continue with breeding the F2 female with an untreated male, and we get the F3 generation. And we get both males and females of the maternal lineage. And we can do the same thing with the paternal lineage by taking the sons here, breeding them with untreated females across generations to get the F3 generation. So the first study I'm going to be talking about is, I'm going to step through the generation. So I've shown you the F1 generation, and I want to talk a little bit about work done by my graduate student, Kritika Krishnan, on the F2 and the F3 generation. So we didn't even know if there would be a phenotype or what that phenotype would be in the F2 generation. So she took her F2 animals, males and females, because of the interest in sex differences, measured body weight, anogenital distance, monitored timing of puberty, did behavioral testing in adulthood, and then euthanasia. So I'm going to summarize a lot of data. She recently published this study in Environmental Health Perspectives last year. But really is kind of an interesting phenotype. And don't worry about the details. There's no quiz. You don't need to know which lineage or which treatment or which sex responded. But what I want you to notice, kind of as just glancing at the slide, is that the effects in the F2 generation of your grandmother being treated with the EDC is lineage dependent. So we see different effects in maternal and paternal lineage. You can see that there. We see that each EDC has its own phenotype. I've just kind of collapsed the summary of the data for the two EDCs together, but in some cases, the vinclozolin lineage or the vinclozolin descended animals have more effects. In other cases, actually, it is more vinclozolin than PCBs, but there are PCB effects as well. And there are differences between the maternal and the paternal lineage. I should note we see more effects in the paternal than we do in the maternal lineage. So we seem to, if there's vulnerability of germ cells, it seems that the male germ cells are more affected potentially than the female germ cells. So what do we see? Just very, very much in brief. We see that in the F2 generation, body weight is increased, so they're heavier. And there is a hypothesis that EDCs increase body weight in exposed individuals. We also see, I didn't show it, but we also see that in the F1 generation, and we see increased body weight in the F2 generation. Age at puberty is a little bit earlier, and I think that is actually consistent with what we see in humans, where there seems to be greater prevalence of early puberty, especially in females, and actually that's what we saw here, was earlier puberty in females. When we looked at USV calls in these animals, we saw some differences in the paternal vinclozolin animals. We looked at mating behavior in the males or in the females, and we saw some differences in the timing of different aspects of male mating behavior. Female lordosis intensity was specifically increased in maternal vinclozolin females. And we also looked at serum hormones, and estradiol and testosterone were decreased by different EDCs in the paternal males. So the summary of all these data, and I'm sorry, I didn't want to overwhelm you with a lot of graphs. So if anybody wants these slides, I'd be happy to give it to them, or we have published the paper. But there are sex, lineage, and also chemical specific effects in the F2 generation that were exposed as germ cells. Okay, so I'm going to now step from the F2 to the F3 generation. And so what Critica did here was to take some of the F3 generation animals that were born through that prior breeding protocol that I showed you through maternal or paternal, and do some of the same types of endpoints. But she was really interested in kind of this crossover relationship between the F2 and the F3, because the F2 mothers interact with their F3 pups. And so one of the factors that changes behavior in rats, and presumably people also, are maternal interactions with their pups. And so Critica took some of those F2 animals and, after breeding them, quantified maternal behavior, looking at things like nursing, arched back nursing, which looks kind of like this, hunched over the babies, passive nursing, licking and grooming of the pups. These are normal maternal behaviors in rats. And she also looked at behaviors within the pups themselves. So that's mother to pup. And then in the pups, she quantified their USV calls, because pups cry when you take them away from the mother, and we can record those, and their behaviors in adulthood. And we also took the brains of these animals and were able to do a little bit of gene expression work. Okay, so to summarize those results, those were just published. So what we found were that the F2 maternal behavior to the F3 pups was significantly affected in the EDC animals, with time nursing, time on the neck, or time on the nest, licking and grooming altered in the EDC animals. the pup calls, their number of calls and the duration of calls when they were recorded on P6 were significantly affected in the EDC lineage animals. And behaviors as adults of the F3 animals in the open field and the light dark box test were also affected. Now, most of these effects were seen in males. I didn't give you any details here, but we had more effects in males. We had about equal effects of vinclozolin and PCBs. So because the effects were mainly in males, she looked at a small number of qPCR reactions of androgen receptor, estrogen receptor, and progesterone receptor, and found that AR and ER alpha in the preoptic area and ER alpha and progesterone receptor in the ventromedial nucleus in the F3 animals as adults was affected by the prenatal treatments. So we think that what's going on is that there's a combination of the maternal behavior along with the EDC lineage that will together determine the F3 phenotype of behavior and neuromolecular changes. So let me spend a little bit more time talking about that, because I'm going to tell you one more story that kind of that we were doing at the same time, but really nicely dovetails with the work that Critica was doing. So how does information get from an exposed dam, a pregnant dam, to her pups, to her grandchildren, to her great-grandchildren? This could also, by the way, be a male. We know there's also paternal transmission. But in my lab, we just expose the pregnant females. Now, what I've shown you is that, let me differentiate between these two concepts. So if the germ cells are affected or changed, the F2 germ cells, and that's all that's the only change there was, then there does not need to be any further exposure or any further manipulation to see an effect in the F3 generation. And there has been work just looking simply at one hit back here, taking things to the F3 generation and seeing a phenotype. But we also need to consider the context, because in the real world, we're constantly exposed to endocrine disruptors. So, maybe I'm exposed to something now that I would be exposing offspring to, but if my offspring live in the same place, and they're also being exposed, and then their offspring might continue to be exposed. So that's depicted here as these multiple hits, because we're constantly being hit, and it's impossible to know if an effect in the F3 generation is due to a germline epigenetic effect as opposed to just the continued presence of that insult. And Critica's work also underscores the point that there are other aspects of context, for example, maternal behavior. So maybe you only have one hit, and maybe there isn't an epigenetic effect, and maybe that chemical isn't there anymore because you moved. But if the maternal behavior has been perturbed, then you're still going to see something in future generations if that is propagated across generations. So we decided to try to look at this question just focusing on the germline side of things in the next study I'm about to tell you. And so what we did in that study was we did the same thing as we did before. We exposed animals to either the vehicle, the PCBs, or the vinclozolin to the F0 pregnant dam. And in that study, that study was only on males. We took male F1 offspring, and we did paternal breeding, so we also bred them down to the F3, and we took F3 male generation offspring, and we looked at one epigenetic modification, which was DNA methylation. And the reason we did that, actually there were a couple of reasons. One was that the original work looking at transgenerational effects of EDCs that was published by Mike Skinner from Washington State had shown that vinclozolin prenatal exposure caused changes to sperm methylation across 3 generations. but he did really, really high dose vinclozolin exposure, basically toxic exposures. So we wanted to do this kind of study using a much more realistic kind of dose. I should note, by the way, that the doses that that we use to treat our rats are probably pretty similar to the kinds of doses that we're exposed to out in the real world and the kind of wildlife exposures that are happening right now. So we're trying to model something in a reasonable range. Now, methylation is an attractive endpoint because if you think about what's going on within the F1 fetus with the F2 germ cells, during embryonic development from day 7 through birth in a rat, or this may be a mouse actually, but during fetal development, the germ cells start out being highly methylated with methyl groups added as this epigenetic modification. And they become, during the course of germline development, completely demethylated. So the entire germline is demethylated by about mid-gestation. Now, as gestation continues in males, methylation patterns begin to get reestablished. And so you can see that shown here within the prospermatogonia, so that by the time of birth, whatever the methylation pattern was has now been established within the male germ cells. That is different in the females, and actually almost nobody studies females. We just got funded to do this work in females, so you can invite me back in 5 years, and I'll tell you what's going on in the females, but I'm going to talk about the males today. The females don't undergo that remethylation process. Here. They don't do it until they start going through puberty because the oocytes are in meiotic arrest here. So they don't complete that remethylation until much later. And that may actually explain a lot of sex differences, why we, across generations and even within exposed animals. So our graduate student, Ross Gillette, decided to do a study looking at methylation in sperm of the F1 and the F3 males, and also the brain. And the reason that the brain was kind of a unique endpoint for this study is that we have behavioral changes in the F3 generation. Presumably, those changes are getting there through some sort of change to the germ cells that in some way are being manifested as a change in a somatic tissue like the nervous system. We do not know how that happens. If anybody has any information on that or ideas, I'd love to hear it. What we wanted to do was simply to compare across generations, F1 and F3 generation, DNA methylation patterns, and then also compare across tissues within the same animals. So along with the sperm, we also chose 2 brain regions, the central amygdala and CA3 of the hippocampus. I haven't talked much about these regions before. We have looked at them in some of our endocrine disrupting studies, but we picked them because we know that in F3 generation animals, we see disruption of behaviors associated with these structures related to things like memory and anxiety and stress. And we had also done some transcriptomics work showing that these regions have a lot of differences caused by endocrine disruptors. So what we did was a sequencing technique that's called RRBS, or reduced representation by sulfite sequencing of both the sperm and the brain. And that will selectively identify sites that are CPG rich with the idea that they're probably CPG islands. And then we can sequence it and determine differences between the different treatment groups within and across generations and within and across tissues. So I'm going to show you 3 slides. They all kind of look the same. They kind of all look like this. And so let me just show you this one in a little bit more detail. So what this shows is CPG methylation in promoters in sperm with the treatment groups shown relative to vehicles. So the heat map is hierarchical cluster analysis of the DNA methylation patterns. Red is hypermethylated, blue is hypomethylated. In the F1 generation and vinclozolin compared to vehicle, and then in the F3 generation of the two treatments compared to vehicle. When you actually add up the number of CpG islands that are hyper or hypomethylated compared to vehicle, there were more hypermethylations than hypomethylations in all the tissues. So overall, there were about 200 to 300 differentially methylated sites in the different treatments and the different generations. We also looked at what classes were predominantly affected, and it was nucleic acid binding, transcription factors, some enzymes, receptors, signaling. So molecules that kind of, I guess, they made sense to us. Maybe at least they make sense to us in the brain, but as I'll show you in a minute, even though this is sperm, we had a lot of similarities between the brain and the sperm. So when I show you the next two, oops, wrong way. Here's the CA3 of the hippocampus. Methylation is a little bit more equal, hyper and hypo, except in the F3 PCB group where it's predominantly hypermethylated. Again, nucleic acid binding, transcription factors, receptors, signaling. And then finally, the central amygdala, similar pattern. Not as many differentially methylated sites, but again, largely hypermethylated, transcription factors, nucleic acid binding, et cetera. So this is really hard to interpret. So what we wanted to do was to see whether there were actually any commonalities. And there were. And we've just published this paper recently. If you're interested in looking for your favorite gene, they're listed there. But what Ross did that I thought was pretty clever was to make a gene cloud. And what this shows is the overlap of the commonalities within each treatment across the different tissues. So and this is in the F3 generation. And we have this information for the other generations, but actually the results were pretty similar for the F1 generation. So for the PCBs in sperm, CA3 and central amygdala, these were the differentially regulated genes. This is the small cloud of genes that are in common between the two brain regions or between the brain regions in sperm. And these are the genes that were in common amongst all of them. And you can see that all of the common genes, and the size means the number of CpG islands that were identified. And there was a type of gene that came up called pRNA, which I never even had heard of before, but they are promoter-associated RNAs, and they have the function of silencing ribosomal RNA, which means they have the ability to affect translation, which to me is really interesting when I think about my gene and protein data. The gene expression and the protein expression data often don't go in the same direction. So it may be that we're affecting some sort of translational or post-translational process. And the promoter-associated RNAs were all hypermethylated in brain, in sperm, and in both treatment groups. So that is the one commonality that I think is really interesting. I've done a little bit of reading. There's not a whole lot out there on promoter-associated RNAs, but I think they're a really interesting target that we hope to be able to pursue. Many of the affected CpG islands were in intergenic regions associated with non-coding RNAs, among which were these promoter-associated RNAs. Promoter-associated RNAs are sensitive to methylation status of nearby CpG islands, with increased methylation decreasing their expression. And overall, we identified 6 of them that were identified across all tissues and generations, and by both EDCs as well. So we still haven't answered the question of how are heritable traits carried forward into future generations, and how might environmental chemicals produce deviations from the course of events. But that's I guess the goal of the next however many years that I'll be doing this research. And again, I should mention that we plan to be able to do a study similar to this in the females. It's harder to do it in the females because it's very hard to collect, germ cells in the females, but we have a collaborator who can do that. Okay, so I'm going to try to give you a take-home message. So what I've shown you is that there are direct intergenerational to the F2 and transgenerational to the F3 effects of EDCs, that prenatal EDCs can change hormones, physiology, behavior, gene and protein expression in exposed animals and transgenerationally. The outcomes are almost always sexually dimorphic. We see different outcomes across lineages. That is to be expected because of different sort of stage of being of when the exposures happen. And they're very strongly influenced by the maternal or the paternal lineage. We believe both context and germline epigenetic modifications contribute to the transgenerational effects. And I ended by showing you that epigenetic profiles of DNA methylation in sperm, central amygdala, and CA3 of hippocampus are affected by the EDCs in at least the F1 and the F3 generation, with similar hypermethylation of promoter-associated RNAs. So we are the combination of the past and present exposures that will determine the future. Okay, so let me end here by thanking the people who did all of the work, the NIH for funding, our rats, David Cruz, it looks like he's out from outer space, who has been my collaborator on much of this work, all of my students and people in the lab who have just been amazing, oops, and here they are. We have usually about a dozen undergraduates in the lab at any time, and they have been phenomenal at doing this, helping with raising animals and taking care of them and preparing tissues of animals across multiple generations. So I'll stop there. I think, can we ask questions? I think maybe, if there's time.
Thank you. That was a very interesting presentation. I'm not a scientist or a doctor, but I'm very interested in the area generally, as someone with more of an environmental background. I'm curious to know, how can we protect ourselves, and how concerned should we be about wine? And does it make a difference if one is consuming wine from Europe or wine that's organic, or is red versus white? I mean, is there any advice you can give us?
Well, I don't know if I can tell you what brand of wine to drink. I can give you some general advice. I don't want to get specific about wine. I mean, organic wine is never as good as non-organic wine, but if you can stand to drink it, then hopefully it has not been treated with fungicides. Of course, when you see the word organic on food, that can actually mean a lot of different things, but I would certainly look for something that's pesticide-free, or maybe go to local wineries and try to find out what they treat things with. So I think, in terms of eating and diet, shopping locally, if you can, is a good idea. If you can actually get things from the farmers where you can get information about how things were raised. I generally recommend trying to avoid hormones in meat or any kind of treatments in meat, because that can actually get in your body and not get broken down as much as some other, growth hormone can break down, but steroid hormones don't. To wash fruit and vegetables, to get pesticides off. You can't wash your wine, but you can wash your apples. And to try, I know I talked about this with some people earlier, try to avoid canned foods, try to avoid packaged foods. If you are microwaving, don't microwave in plastic.
That was great. I really enjoyed it. I had a question is, if you stress your animals, do you bring out more of a phenotype? For example, you showed that there was increased weight gain with some of the treatments, and I was wondering, well, if you feed the animals a high-fat diet, does that exacerbate the weight gain, or do you lose the phenotype?
So we haven't done the high-fat diet, but there are other people giving high-fat diet. Typically, it exacerbates. We actually subject our animals. We did some transgenerational work where we took the F3 descendants of the treated animals and then ran them through a restraint stress protocol. So in other words, your ancestry, your ancestral EDCs, and how do they interact with stress in your own lifetime? And they had a pretty profound effect. It wasn't entirely predictable. It wasn't just that if you descended from vinclozolin, your stress response was worse, but it ended up creating a complex phenotype that was dependent upon the interaction of the EDC heritage, I guess, and the stress that the animals were subjected to. So we think it's just changing trajectories, changing norms of reaction that result more often than not in some sort of adversity or adverse outcome.
Andrea, thanks. That was great. I have a question about the, sort of the transgenerational and the concept that it may be due to the maternal, the maternal influence. I know that with mice, you can switch the litters around so that you could take the mice from an affected female or an exposed mother to an unexposed mother. I don't know if rats have worked with rats.
You can do that.
So have you or have others tried that to see?
Separate out
the maternal effect?
So I did it once, but it wasn't to separate the maternal effects. Actually, I cross-fostered just to even out litters, but they were same treatment. So the problem is that the EDCs often will pass to the young through lactation. So if you and that probably is a very relevant mode of exposure. So we usually like to keep the animals with their mothers. But Emily Rissman, what she does, she uses mice and she treats her animals prenatally, and then she cross-fosters them all to untreated animals. So that way she can actually isolate she can just look at the prenatal exposure and then look at the outcomes. And they still have lots of effects.
So what's the rats versus mice? Are there advantages
Right.
one versus the other?
I think rats are a lot smarter. And it's a lot easier to run them through the behaviors. Yeah, they're very, very cooperative for the behaviors, bigger brains, bigger blood volume. And since this is not, a specific gene focused, I mean, I'm interested in the genes, but my questions, I don't need to do a knockout. And actually now if I need to do a knockout, now I can do it in rats. So, yeah.
This was very exciting. But you partly answered my question about continued exposure through lactation. So you really are not separating out transient exposure, which was primarily the design which I conceived in your initial talk. But this actually imposes another confounding factor that all the pups don't get the same amount of mother's milk. And so then why would you not actually always go to surrogate moms? So I would imagine that actually you would want to take the pups and have timed pregnant females ready to be the surrogate moms to truly, just really stop. Because this continued exposure question that you yourself addressed to almost like, does your vivarium actually have BPA-free bottles of water, right? Which could be a really confounding factor all along the line.
Right. Well, we use glass bottles, so that,
That's
reverse osmosis water. So that's a good question. And in all honesty, the doubling the animal colony to be able to cross-foster, to do these transgenerational studies, where as you're breeding, you're expanding kind of exponentially. It just wasn't logistically possible.
You just need an extra mom, right? You don't keep the litter of the controlled mother, so you only have a foster mom, which is time.
Right, yeah, sure, I guess we could do that. It would be a lot of breeding and killing, but we also, by the way, do cull our litters to the same size. So we do control, so as far as how much milk, hopefully, it should be around the same. We typically cull our litters to, depending upon the study, either 4 and 4 males and females or 5 and 5 males and females, or as close as possible. But there's every possibility that the EDC affected the mother's brain and affected her maternal behavior. So I get that it would be a good experiment to do that.
Going back to the first part of the talk, you mentioned early on that the doses that you're giving are comparable to what humans will be exposed to. Can you just say a little bit more about how that's measured and how one estimates the exposure in humans, and how you would determine whether a rat or a mouse was getting an equivalent dose?
Right.
So it's very important to interpret
Yeah.
how seriously we should take this.
So we didn't measure the chemicals in our own lab. I actually have a student now, I'm trying to get him to do that, because people want to know exactly how much are the animals exposed. And for me, actually, the important question is what's getting into the brain and how long is it lasting? So I think that would be a great study. And actually, nobody's ever done that. There has been a little bit of PCB work and also vinclozolin work looking at, administering these or other doses to pregnant animals and then measuring how much is in the blood or how much is in tissues later, including a little bit of work on brain. And so the amounts that they're giving in those studies where they did that work are similar to what we're using, and we're using the amount that we're using based on those studies. And as far as the human work is concerned, the tissue concentrations in the rats are similar to what we see in like the NHANES database, looking at the NHANES database, which is a CDC, it's the National Health and Nutrition and Environment, what's E, survey. Big project to bank samples, to get information about people. This database, they've been collecting tissues now for like 30 years, I think. And so they have a tremendous amount of information about human body burden. So how much of these chemicals, their prevalence, what tissues they're expressed in, their concentrations. And so we tried to cross-reference what is found in people from that NHANES database, because it is tens or maybe like 30, 40,000 people. And what we know about the exposures to rats, trying to model that.
Does it vary very widely between
Yeah, it does.
And are you going towards the upper end or the median, or
Yeah, I don't think I can answer that because I haven't measured it. I mean, I like to think I'm somewhere in the, like the average person, whoever that is. But I can't say that for certain. Thank you.